Good morning, ladies and gentlemen. My name is Brian Diaz, and this past summer I worked under the mentorship of Jasmine Yang in Dr. Del Dorscheid's lab in Vancouver, Canada, on a project entitled The Characterization of Glycalization Patterns in the Airway Epithelium of Asthmatics. Now, to begin, I will talk a little bit about asthma. It is a global health problem that affects nearly 300 million people worldwide and is the #1 cause of chronic ER visits. Now, the molecular mechanisms that lead to the morphological changes seen within the airway epithelium of asthmatics are poorly understood, but we know the symptoms quite well. And these include reversible airflow obstruction caused by bronchoconstriction and the filling of the airway lumen space by mucus caused by overactive submucosal glands. There's also a thickening of the basement membrane and muscle hypertrophy, which also contributes to this bronchoconstriction. There's also excessive epithelial damage, which is caused by dysregulated repair mechanisms. Now, the epithelium is an interesting target in looking at the causes of these morphological changes because it is the first part of the airway that is in contact with environmental pathogens and other things that could damage the airway. And because these dysregulated repair mechanisms lead to constant inflammation within the airway and just exacerbate the issue. Now, to understand this dysregulated repair mechanism, we must understand normal repair mechanisms and the importance that carbohydrates play in normal epithelial repair. After a section of the epithelium is wounded, the cells in the epithelium express glycosylated proteins, and the patterns which these proteins have carbohydrates attached to them decide what they do and play a role in their function. So after damage, the surrounding cells spread over to the site of injury and proliferate and then differentiate into the different cell types seen within the epithelium. Now, these normal repair mechanisms are not present within the airway epithelium of asthmatics, and we think it may be because of the carbohydrates within the airway are not expressed properly. Now, we did a previous experiment that characterized the glycalization patterns within the normal airway epithelium. The way we did this was with carbohydrate-binding proteins called lectins. And for our negative control, we incubated our lectins with their corresponding competing sugars, which are seen here with no positive staining, which ensured binding specificity. And we can see that in these pictures that there are cell type specific staining. Here we see the basal cells stained, and here we see ciliated cells stained. There are also goblet cells, epithelial cells, there's the basement membrane, and glandular cells. And all these cells were taken into consideration after our experiment was done to do our semi-quantitative analysis. We also did another experiment prior to my arrival in Vancouver that showed that the expression of these lectins are dynamic during epithelial repair. On the top left, we have an uninjured guinea pig trachea, which showed no positive staining for carbohydrates. And we injured a guinea pig trachea and did lectin histochemistry on the injured trachea to show positive staining for these carbohydrates during the repair. And six hours post-injury, we see very intense staining in the repairing epithelial cells. And this staining changes all the way up until I, where we finally see a decrease in our signal, or a weaker stain in these cells. We have also done another previous experiment showing that certain carbohydrates are necessary for normal repair. One of these carbohydrates is called fructose. We treated a confluent monolayer of normal human epithelial cells with alpha-fucosidase to remove these fructose residues from the epithelium. And to ensure that we removed this carbohydrate, we stained them, and it showed no positive staining. And here we see positive staining for fructose in our confluent monolayer. Now, we treated these confluent monolayers with varying concentrations of this enzyme, which degrades as carbohydrate. And we see that with higher concentrations of this enzyme, very little repair is seen 24 hours, well as with lower concentrations. And with our control, the wounded area decreases much more rapidly than with the highest concentration of our enzyme, showing that this specific carbohydrate is necessary for normal repair. And because of these previous experiments, we hypothesized that the expression of carbohydrate residues are important for normal airway repair and function. So we believe that the glycolization patterns within the airway epithelium of asthmatics might be different, and these differences may lead to the morphological changes and the inability to repair itself within asthmatics. To find out whether there are differences in glycolization patterns, we first had to optimize lectin histochemical staining to find appropriate concentrations to stain these lung sections. We then use these concentrations to characterize the glycolization profile of normal and asthmatic lung donors. So first, like I said, we optimize the lectin histochemical staining to find appropriate concentrations for each lectin for their corresponding sugar. We then stained asthmatic and non-asthmatic lung tissue sections for these particular carbohydrates. And while this was done, the experimenter was blinded, so they did not know whether they were staining asthmatic or non-asthmatic lung sections. We then did cell-specific semi-quantification. There were 3 blinded judges who graded the intensity of the staining of each cell type based on a scale from 0 to 3, 0 being no staining, 3 being very, very strong staining. This is a list of the various lectins that we used and the corresponding sugars that they bind to. The next few slides will be representative pictures of each of the lectins and a graph of the semi-quantification, or the cell type specific semi-quantification that we did. Here we see JAC in asthmatic and non-asthmatic sections, and we see a statistically significant difference in secretory cells, and in asthmatic secretory cells were stained more heavily than in non-asthmatics. With LCA, we have representative pictures on the left, and ciliated cells were stained. Ciliated and basal epithelial cells were both stained more intensely than in asthmatics and non-asthmatics. With SNA, we have our representative pictures on the left again, and we see that ciliated cells are also stained more intensely in asthmatics than non-asthmatics. In UEA1, we see epithelial cells stained more weakly in asthmatics and non-asthmatics. To conclude, we did find statistically significant differences between the 2 categories that we had, non-asthmatics and asthmatics, although we do not know yet whether these particular differences play a role in the dysfunction that we see within airway of asthmatics. In the future, we would like to complete the optimization of lectin histochemical staining and to use this optimization to continue staining lung sections in asthmatic and non-asthmatic donors. We would also like to quantify the lectin histochemical staining using color segmentation and cell type specific semi-quantification again, and examine the glycolization profile using air-liquid interface cultures of mechanically injured primary airway epithelial cells from normal and asthmatic donors. And this is important because these ALI cultures simulate the natural environment that these cells are found in, with the nutrients being fed from the bottom, which simulates our tissues, and the airway on top. is my acknowledgements. I'd like to thank you for your attention, and now open the floor for any questions.
I'm curious, why did you use the guinea pig? Is it a good model for asthma?
Yes, we've seen similar. Well, it was just to show that these carbohydrates are they change over time, and that it just reinforced the slide that showed the little cartoon picture of the cells migrating over, and in that slide it actually. Maybe I can skip back here. Do I? Nope, that doesn't work. Oh, wow. It just reinforced this slide where, oh, I didn't know I did that. That during these processes, these various glycosylated proteins are expressed on the surface, so that just confirmed it.